Hi, everyone. Thank you for joining us for this MDA Engaged Community webinar on gene therapy. Gene therapy has emerged in the recent years as a treatment option for some neuromuscular disorders, and it is important for the neuromuscular disease community to learn more about this treatment option for themselves or a loved one. My name is Michelle Barrios, and I am the Community Education Specialist at MDA. We are thrilled to have you join us today for this important and educational webinar. The webinar today is part of our larger MDA Engage flagship community event series, which focuses on bringing the neuromuscular disease community together around education and social opportunities. Be sure to visit the MDA Engage section on mda.org for updates on upcoming virtual education events. We are recording today's event, and we will be posting it to the mda.org website for on-demand viewing to ensure that those who are not able to join us live today are able to access this information. Please note that all phone lines have been muted. We will be having a question and answer session towards the end of the webinar. Please be sure to utilize the Q&A window to type in your questions. If you hover over the bottom of your screen, a tray of webinar icons will appear. Click on the Q&A bubble to open the window and enter your questions. You do not need to wait until the Q&A session to chat in your questions. As questions come up along the webinar, please feel free to send those in. Before we begin, I would like to say thank you to our speaker, whom you will meet shortly. I would also like to thank Pfizer for their support of this MDA Engage webinar on gene therapy. Let's review the objectives for today's webinar. Participants will learn what gene therapy is and how it works to treat neuromuscular diseases, discuss what diagnoses have a current or upcoming gene therapy treatment, and review the pros and cons for gene therapy. Now, I would like to introduce our speaker. Dr. Bidichandani has researched the genetic and epigenetic basis of Friedreich's ataxia for over 20 years. He is currently the David L. Boren Professor of Pediatrics and the CHF Claire Gordon Duncan Endowed Chair in Genetics at the University of Oklahoma Health Sciences Center. His research has been funded by the NIH, MDA, and FARA. Dr. Bidichandani currently serves on MDA's Research Advisory Committee and on FARA's Board of Directors, and in the past was MDA's Vice President for Research. He is a recipient of the Stanton L. Young Master Teacher Award from the University of Oklahoma College of Medicine. He has mentored several PhD students and postdoctoral fellows. And with that, I would like to turn over the webinar to Dr. Bidichandani.
So thank you all for joining us for this MDA Engage webinar, as Michelle just told us. Just before we get started, obviously my name is quite a mouthful. You can feel free to call me Sanjay, or as medical students here, call me Dr. B. That's fine as well. So what I will do is talk about gene therapy for neuromuscular diseases. And I just want to remind everybody that, the Muscular Dystrophy Association, despite its name, actually cares for people who have over 40 different neuromuscular diseases, some of which are muscular dystrophies and many of which are not muscular dystrophies, but they're all under this umbrella of neuromuscular diseases. And before I get into specifics of gene therapy for neuromuscular diseases, I just want to spend a few minutes giving you sort of a historical perspective of just development of therapies for neuromuscular diseases, not just gene therapies, but just therapies for neuromuscular diseases. So, we happen to be at a unique time in history. So in the 1980s and 1990s, a lot of the genes that cause neuromuscular diseases were actually identified. And what I mean by that is a lot of our neuromuscular diseases are actually caused by genetic defects, and to identify those genes represents the starting point after which you can start to understand the pathogenesis and what's actually causing the condition to happen. And only then can you start addressing the development of therapies for those conditions. So it was in the 80s and 90s that many of the genes started to be identified. And then in the 90s and 2000s, a lot of work went into trying to understand how that gene defect actually results in the neuromuscular disease. And then, shortly after that, we started developing animal models for the neuromuscular diseases. And once you get animal models, you can start to experiment and try out different therapeutics, which you cannot put into humans. You start them in animal models first, and then develop better therapies that will eventually go into clinical trials. And a landmark timeline was, towards the end of 2008, 2009 onwards, the big pharmaceutical companies started to get interested in the rare diseases. And that really started the acceleration of drug development for neuromuscular diseases. And at the same time, while the genes and the pathogenesis and animal models were being developed, at the same time, a lot of new tools started to enter into the toolbox of geneticists and people interested in developing therapies. So for example, small molecules have become very easy to make now. Gene therapy, as we will talk about, has actually developed into something that is now in the clinic. Protein therapies, oligonucleotides, various different modalities became not only just available, but actually developed into, the technology developed well enough that we could actually consider putting it into human beings as therapeutic interventions for neuromuscular diseases. So both of these things started to converge. And I like to show this slide. I just want to remind everybody, this is not a joke. The point here is that we are now able to fix the neuromuscular disease, treat the neuromuscular disease in most of the animal models of neuromuscular disease. And this is a good news story. What this means is that we understand the pathogenesis well enough, and we have tools that are competent enough to actually intervene in the pathogenesis of at least the animal models. And this represents sort of the good news story that precedes the therapy in the human setting. So while it looks kind of facetious here, this is actually a philosophically deep concept here. That is that we understand these diseases well enough and have the tools to actually intervene to develop therapies. And this also at the same time has resulted in the understanding in medicine in general that we should start thinking about personalized medicine. Now, this is a term that's been going around now for a few years already. The point here is that understanding the genetic and epigenetic information of the person and then tailoring the drug to actually intervene in that genetic, using that genetic information has now become the basis for therapies for not just neuromuscular diseases, but most genetic conditions that are out there. So like I said, I just wanted to give a general idea of how therapies are developed for human diseases, specifically rare diseases like the neuromuscular diseases we're talking about. It really starts with basic research in terms of identifying the genes, figuring out how that gene defect is actually causing the neuromuscular problem, and then developing the animal models and intervening in the pathogenesis in those animal models. But all of that information cannot actually come into the clinic unless you first do some what's called translational research. Now, what this means is you take these molecules that are working in the animal model and you test them for toxicity. You test them to see how they are distributed in the body. Do they get to the brain? Do they get to the muscle? Do they get to the heart? Do they cause toxic effects? What doses are supposed to be used? What's the best way to deliver it to people? So all of that stuff is done by biotech companies in what we now call translational research. And once that is done, that is when you get the permission from the FDA, the Food and Drug Administration, to actually start what's called clinical trials and broadly clinical research, which is where we can start to give it to people to first see, is this molecule safe? Is it well tolerated? And then ask the secondary question as to whether it is actually causing a clinical benefit, and then do much larger studies to eventually get good information about not only safety, but also efficacy. And then you go to the FDA and say, hey guys, can we now do we have enough information to actually bring this out into the clinic and start giving it to people who have neuromuscular diseases? So all of this stuff has now resulted in the approval by the FDA of several drugs that are now actually given to people who have neuromuscular diseases. And you can see here, eteplirsen for Duchenne muscular dystrophy, Spinraza for SMA. And I'm building up to this red box here, which is Zolgensma, which is a gene therapy for SMA. And we'll talk about this in a little bit more detail in a second. But the point that I want to make in this segment of my talk is that a lot of the understanding of neuromuscular diseases from the basic research perspective has now been taken through translational research and into clinical trials to eventually result in FDA-approved drugs that are available for neuromuscular diseases. So what I'd like to do is now pivot and start talking about gene therapy, okay? So here's the heading for this section, which is recessive genetic conditions are good candidates for gene therapy. Quite a mouthful. And I'm cognizant of the fact that we are actually presenting to an audience that is not necessarily a scientific audience. I know many of you follow all of these developments in great detail, but not everybody does. So what I would like to do is just briefly kind of explain what I mean by recessive genetic condition and why is it that these are the good candidates for gene therapy. So just to remind you again, we've got over 40 different neuromuscular diseases, most of which are actually genetic conditions. And what do we mean by that? Okay, so, our body is made up of trillions of cells, okay? But when you think of it as trillions of cells, what you actually have to realize is there are about 200 different types of cells. So you've got your hepatocyte, which is your liver cell. You've got specific kinds of neurons in different parts of the brain. You've got, cardiac cells. All of these cells they represent about 200 different types of cells that make up the human body, okay? And inside every cell, there's a nucleus, and inside that nucleus, we have the code that is required to make up the human body. And that code is actually present in every nucleus of every cell of our body. But what makes each of these 200 different types of cells unique is that they have different cellular programs. They don't use all of the information. They only use part of the information that's present in the nucleus to actually become a liver cell or become a heart cell or become a certain kind of neuron. And what do we mean by this code, right? So this code is actually 25,000 genes that are present in the nucleus. And for now, let's just call these genes protein factories, okay? So we have 25,000 protein factories. And actually, to be more accurate, we have 25,000 pairs of protein factories. So for any one protein, we get a copy that comes from mom, and we get a copy that we get from our dads, and therefore we have a pair of these protein factories. And the whole point of these protein factories or genes is to make proteins. And when you have two functioning genes, you make enough protein for that protein to then be used by the cell to either become a liver cell or a neuron or a muscle cell and so on. Now, occasionally, we have a broken gene or a missing gene. So there's a defect in the gene where it is not functional. So it's not producing what it's supposed to produce, which is a protein. And when that happens, you're making half the amount of protein from that gene pair because one is not working and one is working. It actually doesn't matter which one is broken, mom's copy or dad's copy. You only have half the genes that are working, so you're only making half the proteins. And the good news here is that in this kind of setting, really half of the protein is more than enough. There's a lot of redundancy in our biochemical pathways that half the protein is actually totally compatible with normal gene function and normal cellular function and normal organ function. Now, occasionally you will have somebody who ends up with both copies that are broken or missing. Now, when you have both copies broken or missing, you're going to get a severe deficiency of that protein. And that situation is now not compatible with normal functioning. And we call this condition a recessive condition. Many of you might have heard of genetic conditions like recessive or dominant. This is what we mean by a recessive condition, when both the genes are not active. They are broken or missing, and they're not functioning at the normal level, resulting in a severe deficiency of that protein. And the condition that you get in this setting is called a recessive genetic condition. It's genetic because you are altering gene function, but you're losing the function of both copies. And when you get a genetic condition under that setting, you're set to have a recessive condition. And recessive conditions are good candidates for gene therapy. And I'll come to that in a second. Before I tell you that, let me just tell you what a dominant condition is. Now, remember I told you in some settings, you can get a defect in a gene where the gene is either missing or it's not functioning. But occasionally, you can get a change in a gene where the gene is actually still intact, it's still active, but it's doing an abnormal function, okay? And so it's producing, instead of producing the normal variety of proteins, it actually now starts to produce abnormal proteins. And these abnormal proteins are actually what's causing the problem in these people. And this setting is called a dominant condition. So when you have a defect that is producing abnormal proteins, that is what we call a dominant condition. And dominant conditions, even though they're genetic conditions, these are not obvious candidates for gene therapy. Let me just clarify, there are people coming up with gene therapeutic strategies for even dominant conditions, but these are not obvious candidates, and it's not a straight up, you don't have protein, let's do gene therapy. So to recap, the good candidate for gene therapy is a recessive genetic condition where we have both dad and mom's gene copies either broken or missing, but certainly not functioning at the normal level where we can actually consider them to have, where they're having a deficiency of the protein and they get this recessive condition. So when you think about it conceptually, right? So you don't have a protein or you have very little of that protein. So the obvious question is, why don't we give them the protein, right? That's a therapy. So for instance, we know a lot of people who have diabetes, right? And diabetes, we know, it's not, a neuromuscular condition, but we know people who have diabetes, and these people are missing insulin, for example, and we give them insulin. So this is a protein deficiency, and the therapy is, let's give them insulin. You can do that with growth hormone, you can do that with many enzymes. And so that's one way to actually treat a recessive condition. You don't have proteins, let's give proteins. Now, while conceptually that makes sense and it seems like an easy thing to do, very often what we need actually is for the protein to be present in a very specific compartment of our body. So we want it to get to the motor neurons in our brain or in our spinal cord. And when you just give protein intravenously to somebody, it doesn't always end up in exactly the right place in the right form. And so that's the problem with trying to get protein delivery to work. Okay, so where protein delivery actually works, for example, in the case of Pompe disease, which is one of the conditions that is in the list of MDA's conditions, you can do what's called an enzyme replacement therapy. So they're missing this enzyme called acid alpha glucosidase, and we can actually synthesize that enzyme and give it to people. Now, the problem with the good news is that you're giving them something that they're missing. The bad news is that it doesn't get to all the compartments where it is needed. So it helps the heart condition, doesn't quite help the skeletal muscle condition that well, and it certainly doesn't get through to the brain in large enough quantities for it to work there. And so while it does help some people, it's still not an effective way to get around completely fixing Pompe. And so Pompe now again becomes another candidate for gene therapy. Now, gene therapy actually gets around some of these problems of protein therapy. You're actually giving the gene in a way that it gets to the right place in the body where it will then express the protein, and you will have then corrected the deficiency, except you're correcting it in exactly the compartment where it needs to get corrected. So we're going to see this. This is going to develop. I know some people may be thinking this is not entirely clear. Hopefully, as we go through this, it'll become clearer. And of course, I'll be happy to take questions at the end to address some of the misconceptions or any problems in my explanation. And I think it's important to realize here that we rely a lot on viruses to actually get gene therapy to be effective. What do I mean by that? So here's a kind of generic slide that's showing us, a bunch of different viral infections that we naturally incur as human beings, right? So we can get any of these different types of viral infections. Of course, the famous one right now, everybody's heard of SARS-CoV, which is causing COVID at this point. And so these viruses have a natural ability to go to the lung, to the liver, to the brain, to the eye. And so scientists thought, we don't actually have to even figure out how to get to the right compartment because viruses have already figured this out. Why don't we use the virus itself to piggyback our gene into the virus? And because the virus knows where to go, it will take our gene there and the gene will be expressed there. And that's where we will actually get the protein delivery to happen and let the virus actually accomplish the delivery problem, okay? So the way that is done, okay, is so a virus looks like this. This is kind of a generic virus, and the virus has a genome in it. And the whole concept of a virus, it's a very efficient replication machinery, okay? It gets to the target tissue where it's supposed to get to, it replicates, makes many copies of itself, and it goes to other cells, and it causes a viral disease. Now that's the scary part, okay? And gene therapists said, why don't we modify the genome of the virus, okay, in such a way that it cannot replicate. It can still do what we need it to do, meaning that it can still target the desirable tissues. It can get to the brain, for instance, if we want it to get to the brain, get to the liver, the heart, wherever it is we want it to get to. And there's a whole panoply of different viruses that you could use to say, I wanted to get to the heart, so I'm going to use this virus or this subtype of this virus, right? So you take the virus and you modify its genome so it still maintains the ability to target the desirable tissues, and it can still provide long-term benefit. But what it cannot do is it cannot replicate and it cannot cause disease. So what you do is you gut the viral genome, you take away all the genes that it uses to replicate itself, and in place of that empty space, we actually put a gene factory or a protein factory that we want in there. So we put a gene in there that is missing in one of our neuromuscular diseases, and then we deliver this virus to the person. Now remember, this virus will retain its ability to get to the desirable tissue, but it cannot replicate. So it'll get there, it'll hang about there, and it'll produce whatever this gene is producing in the desirable or beneficial tissue, and it will not replicate and not cause disease. So you're getting the good part of this thing, and you're avoiding some of the bad parts of viral infections. So that's the general concept of viral gene therapy. And here's an example of AAV, adeno-associated viruses, given intravenously. Remember, you're giving it IV to, in this case, this is a mouse model, you're giving it IV and you're seeing very efficient delivery to all parts of the brain. Everywhere you see green color, that's where that gene is actually expressing. And you don't have to figure out how this AAV is actually getting to all of these brain compartments. You just know that I need this gene that is deficient to get to the brain and express there. Well, you use this viral vector that's been gutted, so it cannot replicate, it cannot cause the disease that it would have caused, but it now delivers the beneficial gene to the correct tissues that we want it to be expressed in. Okay, so what we've seen so far is this general concept of development of therapies for neuromuscular diseases. We've seen that many of the neuromuscular diseases neuromuscular diseases are actually genetic defects, but it's the recessive genetic defects, the ones that cause SMA, for example, spinal muscular atrophy, the ones that cause Duchenne muscular dystrophy. These are recessive conditions and natural candidates for gene therapy. And we've also seen that you can give IV or systemic delivery of viral vectors that can actually target the correct tissues and cell types that are needed to be targeted, and they deliver the gene that is missing there without causing some of the side effects of the viral infection itself, which is it doesn't allow the virus to replicate and it doesn't allow it to cause a disease. So let's look at an actual example of spinal muscular atrophy. Okay, now SMA is a genetic condition. It is, you actually get a wide variety of clinical presentations for SMA. They're called type 1, type 2, type 3, and type 4, based on whether you're getting a very early age of onset to intermediate onset to late age onset. I just want to say all of these pictures are from MDA.org. Several of these people you might have actually known. These are, public figures in the MDA sphere. They've been national spokespeople for, research in SMA. I just want to say that, now after many years, it's understood why it is that people who get SMA, so it's the same condition, why it is that you get the early age of onset in about 60% of people, and then a few people will get, a much, much later age of onset. And I'll just tell you that in a second so you'll understand, how it is that a therapy could be developed based on the knowledge of the genetics of this condition. So SMA, the gene for SMA is called SMN, okay? And that's shown in these green boxes here. So all of us who don't have SMA have one green box that we get from our moms and one green box that we get from our dads. Okay, so this is the SMN1 gene, and I'm showing you that same gene factory or protein factory. This is the protein factory that's going to make SMN. SMN is called survival motor neuron. It's very important for the survival of motor neurons, this protein. And so these, as long as you have at least one of these green genes, then you are going to be fine. You're not going to get SMA. Now, in addition to these green genes, we also have some of these backup copies, and these are called SMN2 genes. Now, SMN2 genes, they work at about 5% or 10% the efficiency of these green genes. But the point is, as long as you have green genes, we don't really depend on these blue genes. They're there, that's great. Sometimes they're there at small numbers, sometimes they're there at large numbers. But because we have these green genes, and as long as we have at least one of these green genes, we're not going to get the recessive condition called SMA, okay? For SMA to develop, you're going to actually have to miss both of these green genes. And indeed, individuals with SMA are missing those green genes. Now, when you're missing these green genes, that's when it becomes absolutely critical to figure out, hey, wait a minute, how many of these backup blue genes do we have? Because that's going to determine how severely you're affected or not, right? When you have the green genes, those blue genes are there just as extra SMN content, which you don't really need. But once you're missing both the green genes, now you're dependent on that 5-10% function that comes from these blue backup copies of the SMN1 gene. And when you look at people, they actually have 1, 2, 3, or 4, and sometimes 5 of these backup copies of SMN2. This is the SMN2 gene. And the more backup copies you have, the better off you are. And the more backup copies you have, you end up getting the adult version of SMA. The fewer backup copies you have, you end up getting the most severe, early onset type 1 version of SMA. And unfortunately, the most common presentation of SMA, 60% of people with SMA actually get the the SMA type 1, early onset variety of SMA. And so the question is, you're missing these green genes, right? So why don't we just deliver SMN to people? Well, the problem with giving SMN protein is that it will not get to the motor neurons in an efficient manner, and that's not going to be a way to fix SMA. So the next question is, okay, we can't get the protein there. How about we do gene therapy, right? So how about we take this green gene and we put it into an AAV vector, a viral vector that's been gutted. So this vector cannot replicate, it cannot cause any diseases, but it can take this SMN1 gene. And because I showed you the AAV vector can go to the brain, it can actually go to the motor neurons. And so why don't we try delivering SMN1 using this strategy, as opposed to protein delivery, we do gene therapy. And so that's exactly what was done in a gene therapy trial. And this trial was conducted a few years ago by a company called Avexis, which eventually merged with Novartis. And so you've got, this survival motor neuron, or SMN, which was put into an AAV9 vector. And remember, it was given as a single IV injection, right? So you're not coming close to the brain, but the virus knows how to get to the motor neurons, and actually did get to the motor neurons. And here is the result that was seen in the clinical trial. And let me just tell you what you're seeing in this graph. So in this graph, you are seeing on the y-axis, so this vertical axis, you're seeing how well the person, in this case, a child with SMA type 1, how well this person performs on what's a motor function test. So this test is called a CHOP Intend score. It's a composite score of motor function, how well your muscles are actually working. And here's a historical threshold, this number 40 here, that's marking where no SMA type 1 child had ever crossed before in a natural history evaluation of kids with SMA type 1. You just would not go above 40. And these are all people who got the gene therapy. And notice almost all of them, except this one patient here, almost all of them actually surpassed that number 40 and crossed that. So they got enough motor function from that single IV injection of AAV that delivered that green gene, SMN1, to motor neurons that actually caused not only improved motor function as shown by this Intend score, but also superior achievement of motor milestones, longer survival, better motor function in general, and crossed what in history, in natural history, no SMA type 1 patient had ever crossed. And again, this is quite a remarkable development. And based on this and other studies, the FDA approved this drug, and this drug has a name now, Zolgensma, which is essentially SMN type 1 gene delivered by AAV9 by a single IV administration, and it's been approved for children under 2 years of age who have SMN1 genes missing. So they're missing both the green genes and they have spinal muscular atrophy because of this. And this is a drug that's given now in the clinic. It's given one time, just one IV injection, pretty magical if you think about it. Because of that, now it is almost unethical to not screen all the babies to see who has SMA. And so now we do what's called newborn screening. And in newborn screen tests, you look for SMA, and if SMA is present, then these kids become candidates for this gene therapy. And then these guys will either never develop SMA or develop a very mild version of SMA, essentially treating this condition that would otherwise have had a pretty dismal prognosis. And today, so this is as of March the 1st, 34 states are actually doing this newborn screening. I'm in the state of Oklahoma. It's shown in gray here. I know that our newborn screening program is actually planning to institute the screen as well. It just got delayed a little bit because of COVID restrictions, but this is going to be coming on fairly soon. So, very soon, all the babies born in the 50 states will actually get screened for SMA. Pretty historic if you think about it in terms of identifying the gene, figuring out the concept of the green genes and the blue genes, figuring out that it needs to get to the motor neurons, and then figuring out that AAV9 can actually do that if the intervention is done early enough and at the right dose, that you can actually cause this kind of historic benefit. So this is the good news story of gene therapy development in neuromuscular diseases. And based on this, we've got lots of optimism that other neuromuscular diseases, specifically recessive conditions, as I explained earlier, would actually become great candidates for gene therapy for neuromuscular diseases. So what I'm going to do is, obviously, for MDA, yes, SMA, great job. Now, what about the other conditions? And so Duchenne muscular dystrophy, which is another recessive condition, that's basically caused because we have a broken dystrophin gene. And so the question is, why don't we do gene therapy for Duchenne muscular dystrophy as well, right? So I'm going to show you what's going on for Duchenne muscular dystrophy. And so just to remind everybody, Duchenne muscular dystrophy, affects about 1 in 3,500 males. It's an X-linked condition. What that means is, males have 1 X chromosome and females have 2 X chromosomes. And the dystrophin gene that is broken in people with Duchenne is actually present on the X chromosome. And in females, because you have 2 genes, you have 2 X chromosomes, if you have 1 gene that's broken, the other one compensates for that broken gene. But in males, you only have the 1 X chromosome, so you only have 1 copy of the dystrophin gene. And if it's broken, you don't have another gene that steps in to compensate for that loss. And so that's the reason why Duchenne muscular dystrophy is mostly seen in males. It can be seen in females. It's a rare occurrence, but it can happen. But typically it is seen in males. And the onset is usually 3 to 5 years of age. It starts to affect the limb girdle areas. It's a progressive condition. And unfortunately, the kids will be dependent on a wheelchair by 12 years of age. And so when you look at the muscle, this is what non-Duchenne muscle, so anybody's muscle biopsy who doesn't have a muscular dystrophy, will actually look like this. It's quite a pretty pattern where you see these muscle fibers. But this is what a muscle biopsy looks like from a child with Duchenne muscular dystrophy, where you see a few normal fibers, but mostly it's been, there's a lot of connective tissue and fatty tissue and inflammatory cells that have actually come in place of muscle tissue. And a lot of the muscle has actually been lost and cannot actually function. And this is the basis for why you get muscle weakness in muscular dystrophy. And why is this happening? Well, it's happening because you look at, so here's the normal muscle biopsy that I was talking about. This is what a whole muscle looks like. And this is a bundle of muscle fibers. And here's a single muscle fiber. And the coating of that muscle fiber, which I'm showing you here in cartoon form, has this yellow protein called dystrophin that's hanging from that muscle fiber. And this protein, you can stain for it and see it. So this is what, a person who doesn't have muscular dystrophy, you can see it lights up in white color all around because it's hanging from this muscle fiber. So it stains the muscle fiber. When you do the same thing in Duchenne muscular dystrophy, the non-Duchenne situation, you can see it lighting up, but in Duchenne, these fibers are all silent. You're not seeing any of the dystrophin present there. So we have an absence of dystrophin in Duchenne muscular dystrophy. So again, the same kind of question comes up. Why don't we just give dystrophin? Well, dystrophin, if you were to give dystrophin IV to somebody, the protein is going to get destroyed in the blood circulation, and it certainly won't get to the muscles where it's needed in all the muscle fibers. And so the question then becomes, just like in SMA, why don't we put it into a viral vector, right? And why don't we put the gene into the viral vector? And why don't we pick the right viral vector that will get to all the muscles, and we will have delivered dystrophin that is absent in people with Duchenne, and they will get the dystrophin protein. And there's a little bit of a problem with that strategy, and that is this. Viral vectors tend to have an upper limit as to how much DNA you can put into them. And unfortunately, the dystrophin gene is too big for most of the viral vectors. So it doesn't actually fit well into most of the available viral vectors. So we have viral vectors that can get to the muscle, We just don't have viral vectors that can hold the payload that is this entire dystrophin gene, which is just too big to put into a viral vector and successfully deliver it to the muscle. And so scientists have come up with some strategies. Like I said, here's the AAV vector. It can only hold about half the size of an actual Duchenne, dystrophin gene. And so what scientists have been doing is they've been editing the gene and making it into a micro gene. And there are actually two varieties for this. One is called a mini dystrophin, where they've taken out some of this middle portion to make a smaller version. And then there's a micro dystrophin, which is further edited to make it even smaller and reach the kind of threshold that would make it possible for this payload to actually be inserted into viral vectors that can actually then be delivered in a gene therapy strategy. And Pfizer and Sarepta are two companies. There's a third one also, Solid Biosciences, that's developing mini and micro dystrophin as a delivery strategy. And these are actually in clinical trials presently. And so you have, a version of dystrophin that it won't function absolutely exactly like the full dystrophin. Obviously, you're editing and making it smaller, but the hope is that it will, provide enough functioning for the skeletal muscles that are completely devoid, that are absent a dystrophin today, to actually start to have muscle function that would then result in significant clinical benefit as a gene therapy for Duchenne muscular dystrophy. So I want to add another point here, that is that in addition to Duchenne muscular dystrophy, there are other recessive conditions like limb-girdle muscular dystrophy, and there are several limb-girdle muscular dystrophies now that are also being targeted for gene therapy in the same way that I said, for SMA. Some of these genes are actually small enough that they actually fit pretty well into the viral vectors and are actually in clinical development right now. And so that's, again, sort of, in the same vein as what we saw for SMA. Now, these are just slightly lagging behind that, and hopefully we'll start to see some of these good news stories coming out for our other neuromuscular diseases as well. So I hope in these two examples you can see, obviously a complete success story going all the way from, pathogenesis, from identifying the genes, pathogenesis, animal models, eventually all the way into the clinic, and then seeing how some of these other diseases are also being tackled in the same way, and eventually will result in a clinically meaningful benefit after all of this clinical development is completed for some of the other neuromuscular diseases. So I just want to end by just adding a few, comments about the role of the Muscular Dystrophy Association in accelerating some of these therapies that you're seeing out there in terms of neuromuscular diseases. Now, obviously, many of us are familiar with the MDA care centers or MDA clinics that are located, around the country. Many of these care centers are not only involved in clinical care, but are also involved in clinical research, clinical trials, and even basic research in neuromuscular diseases. And I don't know if people are aware of this, but certainly should be, that the Muscular Dystrophy Association is the largest non-governmental funder of neuromuscular disease research. And this is done by rigorous peer review. So all the money that is raised for research by the Muscular Dystrophy Association is actually goes through rigorous review by the Research Advisory Committee, the Translational Research Advisory Committees, and only the highest ranked applications actually receive funding. And I want to just put this in perspective, because we just talked about SMA having now at least 2 drugs that are approved for clinical use. And there's a lot of optimism in the SMA community because of these 2 drugs that are in the market, one of which is the gene therapy we just talked about. And it's usually the front end, so the people who actually bring the therapy to the clinic that get a lot of the credit, and they should, because they've done a lot of the work at the front end, the back end, to actually make the drug come to the clinic. But just to put things in perspective, MDA historically has put $45 million of research into SMA. I'm talking about only SMA, not Duchenne, not ALS, just SMA. And this money went into, identifying the genes, figuring out the pathogenesis, making the animal models, some of the early work of gene therapy, and some of the early oligonucleotide work that is now in the clinic. A lot of that basic research and translational research happened because of MDA and the National Institutes of Health and other funders that make SMA research work before you can actually then have the bigger pharmaceutical companies that take it finally to market. And I don't want to minimize anybody's role. Everybody's role is important in this whole process. And MDA also does another thing of bringing the scientists and the clinicians together in these conferences to make collaborations happen and for ideas to be shared so that you can actually accelerate the therapy development. And a lot of this comes down to smart partnership, right? So we want the families and stakeholders who have neuromuscular diseases to work with the patient advocacy groups like the MDA, to work with the governmental agencies that are doing this, and to work with the biotech and pharmaceutical industries to actually make a lot of this, drug development to actually happen. And MDA plays a pretty critical role in this, in sort of facilitating this engagement and moving of, drug development into the clinic. I just want to say one more thing, and that is that, MDA recognized many, many years ago that this is going to be a very long process. Remember, they got into this in the 50s and the 60s, and it was only in the 80s and 90s that genes started to get developed. But they had a program in place for a very, very long time to say, yes, we have our clinicians and scientists who are working on neuromuscular diseases today, but who's going to work on it in a decade from now? Who's going to develop the next generation of therapies, even after the first generation therapies have already started to provide some benefit? And MDA has very robust programs to identify people who are either in medical school or in their PhDs or doing fellowship training, or even young faculty members who are in research to actually get engaged in doing neuromuscular disease research, clinical, basic, and translational. I received one of these fellowships when I first came to the United States after doing my PhD in the United Kingdom. I actually came here on a fellowship from, by the MDA that got me started working on Friedreich's ataxia, and I'm still working on Friedreich's ataxia today. And similarly, a lot of the people who are in the neuromuscular disease space started off as fellows or trainees that were funded by the Muscular Dystrophy Association. And you can see the numbers down here at the bottom and the amount of money that's gone historically into actually cultivating this cadre of people that are experts now in neuromuscular disease and are available for not only research, but also the clinical development of therapies. I just want to end here by saying, that in the past, it all seemed like pie in the sky when we were talking about therapies. And now we've got several pies in the oven, some of which have actually come out and they taste really good, like the Zolgensma we talked about in SMA. Some of these pies, they'll not come out as great. Some will burn, some will come out perfectly. And this is a really good news, hopeful time, because a lot of these therapies that have been developed and are successful actually tell us that we should be really optimistic about the ones that are in the pipeline going forward. And yes, there will be hiccups, but we will eventually get there, and there will be therapies for neuromuscular diseases and other genetic conditions. So here's my lab. I work on Friedreich's ataxia. I'm funded, as Michelle told us earlier, by the Muscular Dystrophy Association, but also by FARA. And here are a couple of people who actually have Friedreich's ataxia, which is actually another condition for which gene therapy is being developed. I just didn't bring it here because there's only so many examples I can put into this presentation. So I want to end here by, thanking you, first of all, for inviting me to come and speak. Thanking you for paying attention, and just in recognition that MDA represents hope for the families who have neuromuscular diseases. And I'd like to just say at this point that at least I am very optimistic and hopeful about the development of therapies for neuromuscular diseases over the coming few years. We're going to see many more successes. And so I'll stop here. I'll let Michelle take over. And if there are any questions, I'm happy to take them. Again, no need to struggle with my name. Sanjay or Dr. B is fine. And thank you for your attention.
Thank you, Dr. B, for your awesome presentation. I'm going to stop your share and go back to my screen so we can get to the questions. We already have quite a few in the chat. Let me get to them now. All right, let's find these questions. The first question that I have is, are there any treatments for forms of distal myopathy?
Yeah, so, the conditions that are based on, so, actually, let's bring it up. So 1 second. Oh, this is your screen now, right?
Yes. Do you want to go back
so
to your
we're good.
screen?
We're good. No, I just wanted to say that a lot of the myopathies are mitochondrial or metabolic myopathies, and if they are loss of function, then obvious, meaning the recessive conditions, those are obviously candidates for gene therapy. Now, let me just say that it's not only gene therapies that are being developed for many conditions, right? So we also have protein therapies, oligonucleotides, small molecules. There are many, modalities of therapies that are being developed for neuromuscular diseases. I just want to say that it depends on the specific defect that the person has, and it's the personalized development of the therapy that's actually going to cause the therapy to happen. Now, the reason I bring it up in this manner is to say that some of the low-hanging fruit would be, for example, SMA and Duchenne, where you have a large number of people who have exactly the same defect, right? Like in the case of SMA, we know that the 2 green genes are missing, SMN1 is missing. So that allows for a rare disease model to actually work out, because you're going to have enough people to do a clinical trial and actually have enough people who are at the right stage of disease, where they can actually be included in a clinical trial for outcome measures to be measured. And so what's happening right now is we are actually going through, and I'm sorry to say low-hanging fruit, but that's essentially what is happening, which is that if we have some of the more prevalent, even though they're rare diseases, they're more prevalent compared to some of the rarer, let's say, mitochondrial myopathies, for example, or even Friedreich's ataxia. The condition that I'm working on is rarer than SMA and Duchenne. And so that's the reason why you see a lot of the news and activity in those conditions first. But the same modalities and same outcome measures and things that are being developed for some of the other neuromuscular diseases then automatically benefit the drug development in other neuromuscular diseases. Let me add one more point here, and that is, we take it for granted that it is the is the Food and Drug Administration, the FDA, that's actually going to be approving drugs that come to clinic. And that's true. But you have to recognize that in the last 10 years, the FDA itself has learned how these rare disease programs are actually taken to clinic. They've recognized that for some of the conditions, you have to provide accelerated approval or certain benefits in terms of, you can't have 1,000 patients. You're going to have fewer than that. You can't have, 3 different trials going on, because it's just not possible to, recruit so many people with some of the rare diseases. And so while the scientists are getting better, the clinicians are getting better, the outcome measures are getting better, the tools are getting better, but so are the regulatory bodies. They're getting better as well at kind of recognizing how therapy development should happen for some of the rarer conditions that we care for.
Thank you. Another question that kind of pertains to what you just answered. People are asking if gene therapy will work for ALS and PLS.
Yeah, so let me take ALS. So ALS, remember, is 90% of ALS is what we call sporadic ALS, where it is not a defined genetic condition that's actually causing ALS. There are people who have ALS because they have a specific genetic, variant that's causing the ALS. So, for example, SOD1, you've got the G4C2 repeat expansion that's causing, which we call C9orf ALS. So those forms of ALS then become candidates for some of this personalized medicine type of therapies, those are actually dominant conditions, okay? And some of the clever ways to tackle dominant genetic conditions are actually being applied for ALS. So for example, you can use oligos that will specifically take out the mutant forms of SOD1. There are also editing technologies that are being developed for the G4C2 expansion type, the C9orf type of ALS. So, but that's a subset of ALS. The 90% people who have ALS that don't have a specific genetic variant, there I'm afraid we need a lot more work before we can start thinking about gene therapy type strategies to tackle them.
Thank you, Dr. B. A few more questions, actually more than a few more. Will COVID vaccines have an impact on the potential for future gene therapies?
Yeah, so, let me, now that everybody is paying attention to COVID, this is actually a great opportunity to sort of, let's try to think about this, right? So remember the purpose of gene therapy, okay? When we're talking about, let's say, the SMN protein is missing or the dystrophin protein is missing, the purpose of gene therapy is to develop, is to deliver the missing protein to the right compartment in amounts that will last for a very long time, or at least in a strategy that will allow it to last for a very long time, okay? And also not cause any side effects by replicating and causing other diseases and things like that, okay? That's the purpose of gene therapy for a neuromuscular disease. Now, in contrast, let's think about a vaccine, right? So in a vaccine, for example, for COVID, what we want is to just give enough of an exposure to the body of a piece of the virus for a very short time and in the general circulation where the immune system will kind of catch it. So for instance, in the case of the Johnson & Johnson vaccine that just got approved, or the AstraZeneca vaccine that is being given in other parts of the world, these two vaccines are actually viral vectors. Right? They're using the adenovirus vector, not the adeno-associated virus. It's a different virus, but it's a viral vector. And this vector has the spike protein gene in it. And we're giving it to people, but we're not expecting too much of this viral vector. We're just expecting it to hang about for a little bit of time. We're expecting it to express the spike protein for a little bit of time, just long enough and just at levels enough that the immune system will catch it, will recognize the spike and remember the spike and build an immunity against the spike, but then that protein and that virus and whatever can disappear, it can go away, right? And so we're just putting it into the systemic circulation. We don't expect it to go to any complicated part of the body. We don't expect it to last for very long. We just want it to be there for a short time and give a whiff of that virus so the body can then develop the immune system that can actually target it when it finds the real virus that comes along, right? So you can see the difference here, right? So we're still doing a kind of gene therapy. So I think from a PR perspective, when you tell people that we are going to deliver a virus with a gene, and then you tell people now millions of people have already gotten it, like 30 million people in Great Britain have gotten a viral vector that gave them a whiff of this COVID, virus. It just makes gene therapy seem less freaky. I mean, it's just a thing we do, right? We've got a viral vector, and we put what we want, and we piggyback what we send. But having said that, the technology that we need for gene therapy is a little more stringent and a little bit more technically challenging compared to what's needed for vaccine development. I don't want to, minimize in any way vaccine development. It's a difficult thing, and they've got real problems in terms of scale up. They have to make millions and millions. We don't have to do that, but we have to make it so that it goes to the right compartment, stays there for long enough, expresses at high enough levels, and does not cause a problem. So slightly different goals, but essentially both of them are genes delivered by viral vectors.
Thank you, Dr. B, for that very detailed answer. I had a feeling that that question was going to come up, so I'm glad that you prepared for it. Another question, this is a two-parter. It's asking which forms of muscular dystrophy are recessive, and what is the most common neuromuscular disease that might be a candidate for gene therapy?
Yeah, so the recessive conditions are Duchenne muscular dystrophy, Becker muscular dystrophy, all the limb girdle, most of the limb girdle muscular dystrophies, obviously SMA, Pompe, Friedreich's ataxia. There's some forms of Charcot-Marie tooth that are recessive. Most of the enzyme deficiencies. So when you look at my slide on the right-hand side, there are several that end in ASE, like transaminase, acid maltase, all of these deficiencies. These are all recessive conditions. So all of these conditions are natural candidates for gene therapy. And I think, I understand the sentiment that was expressed in the question. One of the subtexts in there was, which ones are the common ones? And frankly, the common ones are Duchenne muscular dystrophy and SMA, and that's the reason why we are seeing, therapy development in those conditions that are preceding the conditions that will come later on. Now, remember that some of the enzyme deficiencies can be corrected by actually giving the enzyme itself. The problem there is you have to do this all the time, right? You have to keep delivering it as infusions, maybe monthly, maybe weekly. And the frequency will depend on the specific type of enzyme and where it needs to be. So there's a cost factor, and then there's also reactions that the person can develop to those kinds of repeated injections that are given. So that's the reason why we like gene therapy. It's a one and done thing. You give it, and then the person will make it for long enough for the condition to actually be kind of treated. So I would say, many recessive conditions on our list of neuromuscular diseases. The common ones are being targeted already. Some of the rarer ones will benefit from the knowledge that we've gained from, treating some of the commoner ones. But we're talking about a few years. I mean, this is all happening right now. It's all really pretty cool, actually.
Yes, and I know we're past the hour, but Dr. B has given us a few extra minutes of his time, so we will keep asking more questions. I know that you focus on Friedreich's ataxia. Are there any gene therapies? How would they affect a late stage patient as far as improvement? Are there clinical trials allowing wheelchair patients as participants?
Yeah. So first of all, Friedreich's ataxia is a recessive condition. It is caused because we have a deficiency of this protein called frataxin. I just want to say, plug for MDA. MDA was actually one of the initial funders for when the gene was identified. I was actually part of the group that found the gene for Friedreich's ataxia. So I know that, MDA was pivotal in that discovery and some of the later things that also happened. Now, so we understand that you're missing frataxin, right? So there are actually, three main ways to think about how to bring back frataxin that is missing. Now, remember, there are other therapies that are also being developed to correct some of the downstream problems. So you don't have frataxin. What goes awry because you don't have frataxin? Can we fix those things? But in terms of specifically bringing back frataxin, one of the ways is the gene that is silent in people who have Friedreich's ataxia, we can reactivate that gene. So that's one strategy. Okay, that's not a gene therapy strategy necessarily. It's just a way to say, can we deliver something to the cell that will turn the genes that are otherwise there and they otherwise are fine to actually turn them back on and then reverse the Friedreich's ataxia development. And then there's the other one, which is, hey, we don't have frataxin protein. Why don't we give frataxin protein to people? Now, remember, the problem there is, and I said that earlier for SMA and Duchenne, how do we know it'll get to the right place? Well, in the case of Friedreich's ataxia, we need frataxin to get to the mitochondria, which is one specific part of the cell. And so the development of TAT frataxin, which is a cell-penetrating mitochondrial targeting form of frataxin, is being developed in clinical trials presently. Okay, so this is not gene therapy. is actually a way to say, you don't have frataxin, we're going to give you frataxin. Hey, wait a minute, frataxin needs to get to this part. Oh, we're going to engineer it so that this version of frataxin will try its best to get to the right part of the cell, right? Then there's the third version, which is very similar to what we are seeing here with SMA, is using AAV vectors, again, as a way to get to the cells that are deficient in frataxin, that are actually suffering because they're deficient in frataxin. The issue with Friedreich's ataxia is that we have two main compartments for that need frataxin. So one is inside the brain, so in the cerebellum, in the spinal cord. And typically when you're targeting inside the brain, you're choosing to go there and you're choosing not to go to other parts of the body. And we also need the heart and perhaps even all the muscles of our body to also get targeted. And most of the vectors, they're not great at doing both compartments. You have to almost pick, am I going for the heart skeletal muscle? Am I going for the deeper brain structures? And so scientists are trying to find viral vectors that can target both of these with decent enough efficiency that they can then use a one delivery strategy. They're also looking at ways to say, we'll give two separate injections. One will go in the brain and one will go to the heart and the skeletal muscle. That has certain logistical challenges that are having to be addressed. So there's that aspect to it. Now, the question of clinical trials, there are many clinical trials, and actually let's not minimize the importance of clinical studies. There are many studies that are going on, which is because unless we understand, for most rare diseases, there's not enough knowledge that we have to start with about the natural history of the conditions, so we can have enough information to develop good outcome measures to actually use for drug development. So when you have a drug, to test whether it's working or not, most people might think, hey, that's an obvious thing. It's not actually obvious for rare diseases. We actually have to do a lot of clinical studies to figure out great outcome measures that can be used for determining whether a therapy is working or not. And the outcome measure can be a functional one, which is a person is telling you, or a person is able to do more things in terms of a functional outcome, but it can be a biomarker. You can measure things in the blood of a person or by imaging, CT scans, MRI scans, that sort of thing, or echocardiograms, and actually see, is there an effect happening? And a lot of people who have rare diseases are involved in clinical studies and are actually participating, maybe not directly, but certainly in a major way in terms of helping develop the tools that are needed for therapy development. So I would not just say clinical trials, I would say clinical studies are absolutely critical in terms of contributing to development of therapies. And then of course you have clinical trials themselves, which can be early stage, right? So phase 1 trials, you're not looking for efficacy, you're just looking for safety there, right? So that's another place to get involved. And then of course, as phase 2 or pivotal trials start to be developed. Now, functional measures, tend to, in not every case, but in some cases, tend to exclude people who have lost certain functions. And so the subtext of your question about involving people with wheelchairs, if there's an imaging biomarker that's going to be read, of course, people in wheelchairs can participate in that. But if the outcome measure of a particular trial is a certain exercise parameter or a walking test, then of course the ability to walk and conduct the exercise becomes an excluding factor for some people and I'm trying to be general here because I'm not talking to a specific family or a specific person here, trying to be meaningful to the whole Friedreich's ataxia community and actually to the whole neuromuscular disease community as well.
We have limited time, so I will ask one or two more questions. For someone who has a diagnosis of a neuromuscular disease, do you have any suggestions to help them and help pharmaceutical companies and organizations focus on research and trials?
Yeah, so, that's a great question. Let me just say that, getting a diagnosis of a neuromuscular disease is obviously, a life-changing experience. It's life-changing for the person, it's life-changing for the family, for all the loved ones that surround that person. and uh, at that stage, everybody asks a lot of questions of themselves, a lot of questions of their family, and I'm always, hats off to people who are thinking beyond themselves and beyond their families to say, how can I help others who have this condition? How can I help, the people who are developing therapies to actually be able to do that more efficiently? And I meet people like this all the time, and I cannot thank people like, these, obviously the person asking this question is thinking along those same lines. And I would say that one of the best ways to actually contribute is to actively engage with what's called a patient advocacy group. Okay, what do I mean by that? So, let's say I have a genetic condition or a neuromuscular disease, and how do I communicate with people that are out there in terms of, who's doing clinical trials, who are the scientists out there working in that space, or even who are the people raising money to do research in that space? And usually there are, in most neuromuscular disease spaces, there are one or more patient advocacy groups, Muscular Dystrophy Association being one of the biggest ones. There are many other patient advocacy groups depending on which particular condition you're talking about. And engaging with the patient advocacy group suddenly opens up a whole world of people. You'll hear about conferences, you'll hear about clinical trials, clinical studies, volunteer work. You'll hear about, people just wanting you to register in their registry, getting free testing, getting all kinds of, opportunities come up, and you get to decide, do you want to go for a walk and raise money? Do you want to get involved in a registry and clinical study? Do you want to get in a clinical trial? It's totally up to you as to how far you want to take this and how much you're able to do as well. And I can tell you that, I'm obviously very, tightly connected with the Muscular Dystrophy Association and with another patient advocacy group called FARA. And we are highly sensitive to people who are in the neuromuscular disease space and how they can be engaged at multiple levels in terms of making therapies happen eventually. And honestly, without people wanting to do this, doesn't matter how much money pharmaceutical companies bring, it doesn't matter how much money is raised for research, therapy is just not going to happen, right? We will need to have the participation from both sides. And I feel that the patient advocacy group serves as this kind of central, lubricant to this whole process to make this thing move in a smooth manner. Obviously, social media, you're out there, you connect with people. What I would say is, be careful about how you decide which information is to be digested as the correct information for your condition, okay? So, make sure that the source of the information you are, consuming is a reputable source, is one that is using research information, and it's not just, scaring people or just telling people things that are completely based on just personal experience and not based on research and actual, experience of families and people with the condition. That would just be a little caveat I would add in there. How about one more question?
Yeah, one more. Are gene therapy trials geared mainly toward children rather than adults?
Oh, no. So remember that every clinical trial, even whether it's gene therapy or not, has very specific outcome measures, okay? So for instance, if it's Duchenne muscular dystrophy, right? And they say that one of our readouts is going to be, a test that involves people who are walking. Well, obviously, we're going to be taking a population of people who have Duchenne, who have genetically tested as Duchenne, and who want to be involved in the clinical study and a clinical trial and are able to walk not only at the present time, but over the course of the trial that's going to happen. So that's when you'll typically see, they'll say it's going to be kids who are 7 to 9, 8 to 10 years of age, and they have certain specific, stipulations. But that's not always the case. If the clinical readout is going to be a biomarker or another functional measure that doesn't require the person to be, actually walking, then obviously it will change the age requirement for that particular condition. So that's the sort of general way to think about it. But a more specific way to think about it is our conditions are not all involving kids. Myotonic dystrophy is an adult onset condition. ALS is an adult onset condition typically. So obviously, clinical trials in myotonic dystrophy involve adults who have the condition. And so it depends on the natural history of the condition we're talking about that determines the age distribution. And it really comes down to actually inclusion and exclusion criteria. We will say you have to have a genetic test that shows that you have that condition. If you don't have it, we're happy to test to see if you do have it. You have to have certain functional parameters that allow us to then measure a benefit, because otherwise it will hurt the clinical trial if you cannot actually show that there's a beneficial outcome happening. And then it depends on the natural history of the condition of what age group that ends up being. That's the age group that will be involved in the clinical trial.
Thank you so much, Dr. B, for answering all of those questions that we had. I would like to conclude the webinar today by stating that we would love to hear your comments about this webinar. Once the webinar is over, a screen will pop up with a survey. If you have some time, please complete it, as it helps us create programs that you are interested in. Again, thank you to Pfizer for their support of this MDA Engage webinar on gene therapy. If anyone has any questions after this webinar, please feel free to email them to mdaengage@mdausa.org, and we will follow back up with you. Dr. Bidhi Chandani, we sincerely appreciate your time, your expertise, and everything you do for the neuromuscular community. Thank you so much.
You're most welcome. Thank you for the invitation. Yeah.
If you are new to MDA through this program and are diagnosed with one of our 43 diseases under MDA's umbrella, or are a loved one of someone who is diagnosed, we encourage you to stay engaged with MDA. You can do this by visiting mda.org/join and completing a short form. This will keep you up to date on the latest information and education to help keep families informed in the neuromuscular community. This concludes today's MDA Engage webinar, Gene Therapy and Neuromuscular Disease. Thank you very much for attending. Have a great day. Have a great rest of your day. And thank you so much, Dr. B.
Thank you so much. Take care, Michelle. Bye-bye.
Bye-bye.